All right, it is 8 a.m., Monday, June 14th. So let's get started with our third session of Biochemistry 2. So what we're going to do to begin with is we're going to continue our lecture from Wednesday, finish off a few slides there, and those concerned with water. Specifically, we're going to look at acids and bases. Biochemistry, we're really mostly concerned about pHs that are around 7.2. That's about the pH of the cell. So when you're doing experiments in biochemistry, generally you want your system to be around that pH. Not always though, because there are many situations where you want different pHs depending on what type of problem you're going to look at. So we're going to begin with that today. And then after that, we're going to move into our nucleotides. We're going to move into the first biomolecule of the semester. But let's start with our acids and bases. And we're going to start with some definitions. And the last part of the PowerPoint, it looks like it's going to be cut off for you. So let me just rewrite what this says. This says biochemist use pKa instead of Ka. And this says pKa equals log of Ka. So that's what's written there at the bottom. So we just start with some simple definitions, and there's different ways to define an acid and a base. But the definition we're going to go with is that an acid will donate a proton, which will decrease pH, while a base accepts a proton, which causes an increase in pH. And here we have a simple acid-base reaction. And hopefully you remember from general chemistry that when a base, here water, takes on a proton, it makes what's called a conjugate acid. And when acid loses a proton, it makes what's called a conjugate base. And these are called conjugate pairs, right? Now, to determine the strength of an acid, we have this term Ka, the dissociation constant. And if we're looking at this acid reaction, the dissociation constant is you always have your acid at the left and then your conjugate base on the right. So we have our acid dissociating. That's why it's called the dissociation constant, because it's telling us, how much does this acid dissociate? So it's products over reactants. And when we look at this, it should make sense that the higher the value of Ka, the stronger the acid. Because on the numerator, we have conjugate base divided by our acid. So the bigger our numerator is, the more of our acid that dissociated. And when you talk about acid strengths, you actually don't talk about Kas, you talk about pKas. And the reason for that is shown over here on the table on the right. Even though it says just K, these are Kas and these are pKas. And the numbers are a lot easier to talk about when it comes to pKas than it does to Ka. So our strongest acid on our table, oxalic acid, is 5.37 times 10 to the minus 2, or a pKa of 1.27. So it's a lot easier to just say 1.27. And like NH4 is 5.62 times 10 to the minus 10. It's way easier to just say 9.25 when talking about strength. So for pKas, the lower the number, the stronger the acid. So just keep that in mind. And for Kas, it's actually the bigger the number. So they're kind of opposites. One thing to notice, this lowercase p, whenever you see that in chemistry, where it's like lowercase p and then a variable, for example, pH, this lowercase p always means minus log of that variable, right? So pKa means, or sorry, not minus log, just, yeah, it is minus log. I forgot to write that right there, minus log. So pH is minus log of the hydrogen constant, or the hydrogen concentration rather. And pKa is minus log of Ka. So that's what that lowercase p means. And so let's just get comfortable with the idea of acids and conjugate bases. So I just want you to take a minute, and for these structures, A and B, these are acids. I want you to try and come up what the conjugate base looks like. And for C and D, these are bases. I want you to come up what the conjugate acid looks like. So I'm going to give everybody a minute or two to do this. Then I'll come back and answer, as it's really hard for you all to draw on this type of Zoom meeting. So I'll just draw for us. But yeah, take a minute and see if you understand the concept of conjugate acids and conjugate bases. Let me put up the question timer here so I can get an idea of where we're at as a class. If this is in your way, feel free to minimize it, and then just when you're done, reopen it by clicking your polls again. And let me know if you have any questions while doing this. Right, so I did get someone who said no idea how to begin. So just remember, when we're talking about conjugate pairs, an acid loses a proton and forms a conjugate base, while a base takes a proton and becomes a conjugate acid. So that's what we're really testing here. And kind of a secret question that's not explicitly shown, but do you remember from general chemistry which protons are most likely to be acidic? And if I'm adding a proton, where is it most likely to be put? So I'm going to give about 30 more seconds for this. All right. So a lot of us finished but not sure, or got part of the way and not sure. So let's take a look at this. So first we have our acid right here. This is actually an amino acid. This is glycine. Remember, an amino acid is NH3, C double bond O. And if it's an acid, it's going to lose a proton. Here is the only acidic proton. Remember, if it's a hydrogen connected to a carbon, they're not going to be acidic. So the conjugate base is just going to be NH2, no charge now. For B, you have, this is also an amino acid. This is aspartic acid. You have two choices. You have NH3 or COOH. Carboxylic acids, COOH, are much more acidic than amines. Very, very acidic, these groups. So this is where we're going to lose the proton. So that's going to go away. And remember, a proton has a positive 1 charge. So this is going to become COO minus. Since it was COOH neutral, losing that proton makes it minus, negative charge. All right, now we have bases. I want to know where the conjugate acid is going to be. Here we have two choices again. We have a carboxylic group and we have an NH2 group. This one's a little more tricky as we do have a negative charge here, but the proton is most likely going to go to this amine group. These amines are very basic. They like having protons on them. So that's where our proton's going to go. And it's just going to become positively charged now. Remember, protons plus 1 charge. So if it was neutral, it becomes positively charged. Here, very simple group, carboxylic acid, negative charge. The only place where our hydrogen can go is on this oxygen. So I'm going to draw it right where that negative sign was, and this becomes neutral carboxylic acid. So yeah, with an acid, you lose a proton. With a base, you gain a proton to make your conjugate pairs. Any questions about that? All right. If not, we can move on. So we do have a question. Yes.
Can you hear me?
Yep.
Okay, great. So I just wanted to clarify, you said the acids accept the protons?
Acids will donate a proton to make a conjugate base. So that's why for A, it went to NH2. The bases down here, they accept a proton and now they become an acid. The base goes to conjugate acid. And here the acids went to conjugate base. So proton leaves. Here, proton is coming in. Yep. Any other questions? All right. So just to continue on with acids and bases and how that relates to pH, what we're going to be concerned about in biochemistry, like we said last week, is water. The cell is 70% water. And so the biggest acid and base in biochemistry would be water itself. And when you just have pure water, some of that water will go through the process of autoionization. That is, water will split to form H3O plus and OH minus. Here I just have it as H plus and OH minus for shorthand. It really forms these. And what's really interesting about water is that the amount of hydronium and hydroxide at any temperature is equal to a constant. And this constant is called Kw. So at room temperature, when you multiply the amount of hydronium by the amount of hydroxide in a sample of water, it will always equal this number. Always, always, always at 25 degrees C. Different temperatures have different values of Kw. And what this means is that if H and OH are the same amount, we call that neutral. If we have more hydronium than hydroxide, that's acidic. If we have more hydroxide than hydronium, that's basic. And if we take the concentration of H3O plus and do the minus log of that concentration, we call that pH at 25C. So at 25C, if pH is 7, that means that the amount of hydronium and hydroxide are the same amounts. We call that neutral. If pH is below 7, then we have more hydronium than hydroxide. If it's basic, we have more hydroxide than hydronium, right? And this is at 25C. At different temperatures, 7 is not neutral. So if we were like at 80 degrees C, 7 is not neutral. It's some decimal, like 7 point something would be neutral. Also, one cool thing that people don't notice or don't really think about, pH doesn't just go from 1 to 14. pH goes from minus infinity to infinity because it's just minus log of a concentration. But most of the time, we only care about pHs between 1 and 14. So that's your little fun fact for the day. You can have negative pHs. If you have just a pure concentration of acid, that's going to have a negative pH. And what we're going to do is, since this is a class that deals with an experimental science, if any of you are thinking about working in a lab with just a bachelor's degree, what you're going to be doing is making solutions for the most part. So let's make sure we know how to make simple solutions. And so I'm going to do 7A for us, and then I'll give you some time to do 7B. So together, let's calculate the pH of a 200 milliliter solution of pure water that we add 50 milliliters of 1 millimolar hydrochloric acid. All right, so we have a problem, and I like to draw pictures. So I have 200 milliliters of H2O. And to that, I'm going to add 50 milliliters of 1 millimolar HCl. That means my new solution is 250 milliliters, right? And I want to know what is the pH. pH equals what? All right, so this is a dilution equation. And from general chemistry, you learned of an equation to deal with dilutions. Does anyone remember what the dilution equation is? And I just wrote concentration of HCl because that's what we're trying to find here. Dilution equation starts with M1. C1V1 plus C2V2. More or less, it's M1. I do M1. M1V1 equals, oops, V1 equals M2V2. Yep. So what this says is molarity of solution 1 multiplied by volume of solution 1 equals molarity of solution 2 multiplied by volume of solution 2. So this is, as a reminder, it's called the dilution equation. Whenever you're diluting something and you want to find the new molarity, you use this equation. So we have a solution that was 50, M1. We have a solution that's 1 millimolar HCl, and we have 50 milliliters of that. We want to know in our second solution, what's the molarity? We don't know that, but we do know the volume of the second solution. It's 250 milliliters. So 50 milliliters was my V1. My concentration of acid was my M1. My total solution after I mix stuff is my V2. My concentration of HCl was my M2. Right, and so I'm gonna go and solve this. So I'm gonna do solve for M2, which is 50 divided by 250, which is 5, 10, 15, 20, 25, 1/5, which is 0.2. So M2 equals 50 milliliters per millimolar divided by 250 milliliters. Milliliters cancel out. My molarity of acid in my second solution is 0.20 millimolar. All right, before I move on to calculating the pH, let me just pause right there for people to catch up and ask, like, based on the math I've done so far, did I lose anybody? Anyone not sure, you know, how I got my numbers, or not sure if the logic I used, anything at all before I move on to the next part of this calculation? All right, so I imagine we're all good then. So I have a solution that is 0.20 millimolar HCl. So let's calculate the pH. Remember, pH equals minus log of hydronium concentration. HCl, if you remember from Gen Chem, is called a strong acid. A strong acid dissociates 100%. So this whole concentration is going to be hydronium. So pH equals minus log. And this is millimolar. So instead of millimolar, this concentration, again, the brackets means molar. So I have to convert millimolar to molar. So hopefully you remember your prefixes. There is 1,000 millimolar in 1 molar. So lowercase m means 1,000. So millimolar. So this is minus log of 0.20 times 10 to the minus 3. And let me get out my calculator. Equals minus log 0.2 times 10 to the minus 3. The pH of this solution is 3.7. So that is how we can use the dilution equation and pH equation when we are diluting an acid to know what the new pH is going to be. Now in B, I'm going to ask you to use the same equations, but I'm going to be asking it in a different way. And something like this is kind of like how you might get some test questions where it's using the same equations, just slightly asking a different way how to use them. So for B, I want a solution that is 50 milliliters with a pH of 2.10. I have unlimited amount of 10 millimolar HCl. I want to know what volume of 10 millimolar HCl do I need to make this solution. So I'm going to give people like 4 or 5 minutes to try and tackle this. If you have questions while doing this, please let me know. Otherwise, let's figure out some dilution equations here. All right, so I'm going to jump back in here in case we're stuck, because there usually is a little confusion about how to start this problem. So let me delete some of this stuff. And I'm just going to, in case you're floundering a little bit, if you're doing well, you can ignore me. All right. So I want to get a solution, 50 milliliters, pH of 2.10. And I have a solution that's 10 millimolar HCl. I want to know how much volume do I need to make this. So again, this is M1V1 equals M2V2 dilution equation. I'm diluting this acid to make my new solution. So molar, and I'm just going to call this M1, V1, M2, V2. Doesn't matter which solutions 1 and 2, as long as you're constant. So I know that the volume I want to make is 50 milliliters. And I know my molarity of my second solution is 10 millimolar, or 10 times 10 to the minus 3 molar if I want to get out of milli. So the first thing I should figure out is, what is my molarity of solution 1? That's given from pH. Remember, pH equals minus log of hydrogen concentration. Well, my pH is 2.10. That's my pH value. That's equal to minus log hydronium concentration. So I'm trying to solve for this concentration of acid, because I don't know what it is, but I know what my pH is. So let's solve for that. The first thing I'm going to do is move my negative sign over. So negative 2.10 equals log hydrogen concentration. And we talked about this last week when it came to natural logs. I said last week if you have natural log of some variable, the way to get rid of that natural log is to use exponent. They cancel each other out. For log, the way to get rid of a log, if you want to get to a variable, is you do power 10 to each side. Power 10 to each side. So log power 10 or 10 to the power of a log, they cancel each other out. But I have to do it to each side if I want to do a mathematical operation. So I'm doing 10 to the power of negative 2.10. That's my concentration of hydrogen. And my hydrogen concentration, where's my pen? There it is. My hydrogen concentration when doing that is 0.007943 molar. So that is my M1. That's my concentration of acid. 7943. So I have an M1, I have a V1, I have an M2, I don't have a V2, and that's what the question's asking. How much of this do I need? So that's my only unknown. I have one unknown in one equation, so I can solve that. So let's start plugging in. M1, 0.007943, multiplied by the volume I want, 50, equals molarity of volume 2, 10 times 10 to the minus 3, multiplied by what volume do I need? I don't know that yet. So let's solve this. So equals 50. So I'm doing 0.007943 times 50 divided by 10 times 10 raised to the power of minus 3. And my volume of my acid solution that I need is 39.7 milliliters. So I need to take 39.7 milliliters of my acid, and I add that to 10.3 milliliters of water to get my second solution. That's 50 milliliters with a pH of 2.10. Again, these type of calculations are incredibly common in a lab. If you're ever doing lab work with solutions, you're going to be doing an equation like this at some point. So it's very important if you ever think about doing something like that, that you understand these type of equations and how to use them. But before we move on, any questions about anything I did here? Anyone got lost, just want me to re-explain something, or anything at all. Where did I get 10.3 milliliters of water? Sure. So my, whoops, sorry, wrong button. So let me go back to there. So my final solution is 50 milliliters, right? And if I have 30.97 milliliters of my acid that we just calculated, and my final solution is 50 milliliters, the rest of that has to be water. So I just, in my mind, I'm just saying 39.7 plus what equals 50. And that should be 10.3 to get my final volume. So in essence, what you would do in the lab is that you'd measure 39.7 milliliters of your HCl solution, pour that into a new flask, add 10.3 milliliters of water, mix. All right. If there's no other questions, we can move on. And let me just see here. Okay. And we're going to talk about buffers now. A buffer is a solution that is made out of a conjugate pair. So we have three different buffers that are being shown here. We have an ammonia buffer. So the conjugate pair is right here. We have a phosphate buffer, and we have an acetic acid buffer. So a buffer is simply a solution that has both acid and conjugate base. So they have to be conjugate pairs, and they have to be relatively the same concentration, right? And what a buffer does is that they resist pH change. So if you have a solution that has a buffer in it and you add hydrochloric acid, the pH doesn't change all that much because of the buffer. If you add a strong base, same idea. The pH doesn't change all that much. And for a buffer, their range of being an effective buffer is +1 or -1 of their pKa. So for example, ammonia. The pKa of ammonia is roughly 9.2. So ammonia is a good buffer between the pHs of 8.2 and 10.2. Phosphoric acid has a pKa of like 6.8. So it's a good buffer between 5.8 and 7.8. Acetic acid is roughly 4.8. So that's a good buffer between 3.8 and 5.8. So for each buffer, you're usually +1 and -1 of your pKa value. If your solution has a pH that is equal to the pKa of your buffer, you then know that your buffer has 50% acid and 50% base. So if your conjugate pair has the same amount of acid and base, your pH equals your pKa. If you want to know the pH of a buffered solution, and it's not 50-50, we have to use this equation called the Henderson-Hasselbalch equation. What the Henderson-Hasselbalch equation says is that the pH of a buffered solution is equal to the pKa plus the log of concentration of base divided by concentration of acid. So that's Henderson-Hasselbalch. Living organisms have a ton of buffers. The one we'll talk about in biochemistry this semester is the blood buffer, which keeps your blood at a rough pH of like 7.2. It's made from carboxylic acid, basically carbon dioxide in acid form. But we'll talk about that later on. But that's the Henderson-Hasselbalch equation. And so let's get a little practice using the Henderson-Hasselbalch equation this morning. So calculate the pH of a 1 liter solution containing 10 milliliters of 2 millimolar acetic acid and 5 grams of sodium acetate at the pKa of acetic acid is 4.76. So again, let me get started here. So I have a solution. The solution is 1 liter. And to this solution, I added 10. This solution has, rather, 10 milliliters of 2 molar acetic acid. And I also added 5 grams of sodium acetate. Sodium acetate is the conjugate base of acetic acid. So acetic acid is my acid, acetate is my conjugate base. I want to know what the pH is. So for that, we need pKa, we need the concentration of base, and we need the concentration of acid. We actually have everything we need to solve this equation. So again, I'm going to give people a few minutes to think about this and try this. This is a lot of general chemistry again. So try to rack your brains back to when you're in general chemistry to figure out if you can get the concentration of your base and acid. Give people a few minutes to do this, and then I'll come back with the explanation. All right. So lots of moving pieces for this equation. Lots of different things to think about of what to exactly tackle. But let's take a look at this. So I'm just going to try and start putting in numbers here. So pH, what I'm trying to find, because calculate the pH, is equal to pKa. Well, my pK is given 4.76 plus log of concentration of base. So remember, concentration equals moles divided by liters. So I have a weight of base here. I have 5 grams of base. And I know that for every 82 grams of base, I get 1 mole of my base. So I'm taking my weight, dividing by my molecular weight to get amount of moles. So 5 divided by 82 is 0.061. I'm just rounding here. 0.061. So that's my concentration of base. So plus 0.061 divided by, now I got to do my concentration of acid. And this is the third time we're seeing this equation today. M1V1 equals M2V2. So I have 10 milliliters of acid, and it's 2 molar. Whoops, I switched those around. It should have been 2 molar of acid multiplied by 10 milliliters of acid. My molarity in my new solution, I don't know, but my volume is 1 liter or 1,000 milliliters. When doing dilution equation, make sure your volumes are in the same unit, or else it won't work. So this is effectively 20 divided by 1,000. And so my molarity of my acid is 0.02. And now it's just solving that equation. And we see the pH of this equation then is 5.24. And when doing these type of equations, I want you to start asking yourself if the answer you got is an answer you expected. What I mean by that is look at the concentration of acid and base, right? So the concentration of base is 0.061. The concentration of acid is 0.02. So the amount of base we have in the solution is greater than the amount of acid. Therefore, if pH is equal to my pKa, when the amount of base is equal to the amount of acid, if I get into the situation where my base is greater than my acid, I would expect my pH to be greater than pKa. If it's the other situation where my acid is greater than my base, I would expect my pH to be lower than my pKa. So that is just like one of those checks you can do when doing problems like this. Like if you calculated this and your pH wasn't greater than your pKa, then you probably did something wrong. So that's a way you can check yourself. But any questions about this type of buffer problem using the Henderson-Hasselbalch equation? If not, we can move on then. And there's another question here that deals with the Henderson-Hasselbalch equation. For time's sake, I'm going to skip it in class because it's just using the equation again. I would suggest that you try it, and I'll be posting the answer online. But yeah, just for time's sake, I want to move on to our next PowerPoint. So that's the end of the PowerPoint from Wednesday. Let me get the PowerPoint I had up for today. And we are going into chapter 3, moving quite along here in our 2-hour days. And we're going to talk about nucleotides now, our first biomolecule. And here on this slide is just a big list of information. Let me write down what is getting cut off for you. So this is a 3' deoxynucleotide. This is ribose. Oops, not bibose, ribose. And this is deoxyribose. All right, so the nucleotides in our bodies, in all life really, come in 2 flavors. They're purines or they're pyrimidines, right? Purine, double ring, pyrimidine, single ring. And we have 5 common types of nucleotides: A, G, C, U, and T. U is RNA only. T is DNA only. And the RNA and DNA is referring to what sugar we have on our nucleotide. So nucleotides are made out of 2 different, well, really made out of 3 different things. They're made out of a base, a sugar, and usually they have phosphates on them, right? So when we look at the sugar, they come in 2 flavors, a ribose or a deoxyribose. And the only difference is this hydroxyl at the 2' position. So when we talk about the sugars in nucleotides, they are 5 carbon rings. So 1, 2, 3, 4, 5. And they get primes. So 1', 2', 3', 4', 5'. The reason why these are primes is because our bases are numbered and they don't get primes. So to distinguish between, if you're talking about a base atom or a sugar atom, that's when you add the primes. So at the 2' position, we have a hydroxyl. That's for ribose. For deoxyribose, the word deoxy means you lost an oxygen. You lose it at the 2' position, right? So that's the difference between deoxyribose and ribose. And 5' and 3' nucleotides, what that means is the location of a phosphate. So at the 5' end of any nucleotide, you will have a phosphate. So for DNA, right, at the very 5' end, you will have a phosphate hanging out. At the 3' end, you can also have a phosphate. Not always. Sometimes that's just a hydroxyl. But if I'm talking about a 5' ribonucleotide, that means there's a phosphate at the 5' carbon right here. 3' nucleotide means there's a phosphate at the 3' position. What I would like you to do, and I'm going to ask it in some way on the test, is I want you to know the difference between our 5 bases. So you should be able to recognize those 5 bases. You should know the difference between a ribose and a deoxyribose. And you should know what is a 5' and a 3' nucleotide. So just a few things for you to know by our test. So make sure you study this page. And we're just going to start with some naming here. So when we name our nucleotides, we start with the base, so adenosine. And then we say, here is ATP, triphosphate, because we have 3 phosphates. So this would be what ATP looks like. So I have 2 nucleotides here. I want you to take a minute and try and determine what is the systematic name of these nucleotides. I have the chart over here for you. I'm going to give you a minute to do that, and then I will call on 2 people to tell me what their answer was. So take a minute and see if you can figure this out. And the people I'm going to call on, A will be Jude, B will be Lauren. So you can either type in your answer, or if you want to, if you think it'll be easier to speak it, just raise your hand and I will unmute you when you, whenever you get your answer. So for A, we have guanidine diphosphate. That is correct. So our base is a guanine. And then we have 2 phosphates. So this would be GDP, guanidine diphosphate. Is that a phosphate at the end? Yeah. There's an oxygen sticking off here. That is the phosphate. So P, then 3 oxygens. Adenosine monophosphate, yeah, that is either AMP. I guess the more correct name would be cyclic AMP, because the phosphate is forming a ring with our sugar, but it is adenosine monophosphate, 1 phosphate. So yeah, if we have nucleotides and you just have phosphates on them, and they're not connected to any other nucleotides, you just say the name of the base and then tell me how many phosphates it is. So ATP, GDP, and cyclic AMP is shown here. Any questions about that? All right. And that's our intro to what nucleotides look like. So let's actually get to the more interesting thing of what they look like when they are combined. And in general, when nucleotides come together, they form bonds between their 5' and 3' carbon. They form phosphate bonds. All right. So these phosphates negatively charged. That's why DNA, RNA is incredibly negatively charged. And when we read and the body reads, or rather the cell reads DNA and RNA, we always read it the same way. We start at the 5' end and we read it to the 3' end. So here our sequence is AUC CG, right? And so this is a polymer, also called a oligomer. RNA is usually going to be single-stranded, not always, but usually. DNA is usually going to be double-stranded, not always. Here, we're looking at RNA because the hydroxyl is there. So this is RNA. And when talking about DNA, remember Chargaff's rule from general biology. In an organism, the amount of A is going to be equal to the amount of T, and the amount of G is equal to the amount of C. Most organisms follow Chargaff's rule. There's actually organisms that don't, but those are going to be in extremely extreme conditions where they live. But most organisms that we interact with will follow Chargaff's rule. And I'm going to give everybody a minute and just get comfortable with the idea of how nucleotides are connected. So if you were in an in-person class, I would emphasize that I would ask you this question on the test. However, you can't really draw online that well. So you're not going to be asked to draw on the test, but I will most likely, like 95% plus, give you a picture of nucleotides and ask you to tell me what is the sequence of nucleotides. So what I want you to do is just see if you can draw a CU dinucleotide, a single-stranded CU sequence. This is ribose. This is deoxyribose. So I'm going to give you a couple minutes to do some drawing for me. If you have questions, let me know. Otherwise, I'll be back with the answer in a few minutes. All right, so hopefully you had some time to do that. So let's start with our ribose. So we're going to do the 5' position, and at the 5' end, you're going to have a phosphate. oxygen. So then we have CH2. So this is my 5' carbon. Then 1, 2, 3, 4, 5. And then our ring's going to be here. So 4', 3'. Whoops. I am not good at drawing, folks. Let me erase that line. My 4' is here. 4', 3', 2', 1'. So hopefully, as you can see, I'm not an art major. That is really ribose. But then at the 3', that's where we're going to connect to our next our next base. And that's going to be CH2. So here's our 5', 4, 3, 2, 1, oxygen, 4. And then at the bottom of our 3', we're just going to have a hydroxyl. So before I draw the nucleotides, just some things to keep in mind. One, it's RNA. So at the 2' position, we need those hydroxyls, which I just drew. Connecting our nucleotides, they're always going to be connected 3' to 5' through a phosphate. So that's what I have here. At the 5' position, at the very last, you usually have a phosphate. At the 3' position, you can just put an OH. So that's the sugars, and let's draw the bases. The bases are connected to the 1' position. So we start with C. So here's my C. So that's going to be nitrogen. Up here, double bond O, nitrogen, NH2, double bond there, double bond. So that's my C RNA nucleotide. Now my uracil, nitrogen, double bond O, nitrogen, H, double bond O, double bond. So that is my U, and that is a very poor drawing of a RNA dinucleotide. So again, just talking about the bases, what to keep in mind about that is that they connect the 1' position. The oxygen that was there gets removed. So you just have what's called a glycosidic bond. That's what that's called. Connecting your base to your sugar. And they always connect where the X is in this picture. That's showing you where your sugar is located. So that is our CU RNA dinucleotide. Again, I'm not going to ask you to draw this on a test, but be aware I could ask you, like I'll show you a picture of a di or trinucleotide and just ask you to name it for me. So any questions about this? All right. So continuing on about the structure of our nucleotides, DNA is mostly going to be in a double helix. And what a double helix is just two chains that are wrapping around each other with a central axis. And they're anti-parallel, which means if I just stretch my DNA out, one chain is running 5' to 3' in that direction. The other chain is doing it in the opposite direction. Both are right-handed helixes. Don't worry too much about what that exactly means. That just means the way that the loops are facing. DNA itself has two different size grooves. It's called the minor groove and the major groove. It just, in this picture, actually shows it very nicely. The minor groove is kind of more compressed, I would say. It's not really compressed. It's just not as accessible to the nucleotides as the major groove is. There's less space to access nucleotides versus the major groove. The bases are pointing inside of the helix, and on the outside of the helix are your sugars and phosphates. So that's what's interacting with the water, is the sugar and the phosphates. And in a double helix, we're going to have hydrogen bonds, A with T, C with G. That's why Chargaff rule exists. If you have a single-stranded nucleotide, this happens a lot in RNA, that will usually form on itself and make a loop. So RNA, if it's just a single strand, usually doesn't form a double helix. They form a lot of different loop structures that from a single strand just folding on itself. But yeah, that's the basic structure of our DNA double helix. And this is what I was saying on this last image, RNA being single-stranded, has a lot of flexibility, can form a lot of different shapes. And with these different shapes, it allows it to do a lot of different things, binding to molecules, being catalysts. And I mentioned this last week, but that is why we think life started with RNA world theory, is because RNA can be a lot of different shapes and do a lot of interesting things chemically, but it is also genetic information. It could be used as genetic information. So that's why we think all life probably started with RNA. So let's think about DNA itself, right? And here are two questions that I would think are critical thinking questions to see if you really understand the biochemical properties of DNA. So two questions I want you to think about, and let's start with A. Why do the strands of DNA separate more easily if the pH is above 11? So why is it easier to separate double-stranded DNA into single-stranded DNA if I raise the pH? And I'll open up this question to anyone. So if anyone has any ideas, feel free to either raise your hand or put it into chat. Why can I separate DNA easily at high pHs? Breaking of hydrogen bonds, weaker of hydrogen bonds. Yeah, that's correct. So at a high pH, well, first, let's think about DNA itself. So we have double-stranded DNA. And what's holding these two strands together are hydrogen bonds, right? The hydrogen bonds between the bases, as being shown here. When you get to high pHs, that means you're going to start losing protons. And for DNA, when you're losing protons, you're breaking hydrogen bonds. And if you don't have any hydrogen bonds, there's no reason for DNA to stay together, because they only stay together because they're hydrogen bonding. So at high pH, you break hydrogen bonds and DNA separates. So that's why if you want to separate DNA into single-stranded molecules, one technique you can do is just increase the pH. So let's go to B then. B is usually a little more trickier. Historically, when I ask this, people have a little harder time understanding B. So let's say I have DNA and I keep adding NaCl to it. And I'm doing an experiment where I am measuring how much, what temperature do I have to be at to separate double-stranded DNA into single-stranded DNA. This is called melting. So if you melt DNA, you're making it single-stranded. And the question is, why do we see when we increase the salt concentration, the temperature of melting also increases? Does anyone have ideas about that phenomenon? How can we explain that observation based on what we know about DNA, the biochemical structure of DNA? Again, I'll open this to anybody, either in chat or feel free to raise your hand if you have an idea. You assume it has something to do with salt bonding to DNA. Your assumption would be correct. Why does salt binding to DNA make it less likely to form single-stranded DNA? What is it about DNA that could be affected by salt? And to help your thinking along, remember what NaCl is made out of, right? When you put NaCl into water, it forms positively charged sodium, negatively charged chlorine. Why does this make DNA stick together more? Anyone have ideas of why that is? The added negative charges from the Cl. So let's think about that. What is the charge of DNA? What is DNA's overall charge? Is DNA positive, negative, or neutral? Negative, correct. So if DNA is negative, which of these two ions is it most likely to bind to? Is it going to bind to sodium or is it going to bind to chlorine? Sodium, right. So would it require more energy to separate the strands? Because at a higher concentration, I would assume there would be more sodium ions to shield the negative DNA. Yeah, so that's what's going on, right? So let me explain what was just said there. So DNA, incredibly negatively charged. It is just coated with negative charge. And you are bringing two strands together that are incredibly negatively charged. So those strands want to push away from each other, right? Negative things don't want to be around each other. So DNA, the forces of DNA, you have two forces. You actually have three forces. You have repelling forces. You have your negative charge. So the negative charge of DNA is forcing them to repel. And then you have your attraction forces. You have your hydrogen bonds. These are making the DNAs come together. You also have what's called base stacking. That also helps with attraction. Base stacking, and I'm just going to stop sharing here for just a second to put myself on the big camera. Base stacking in DNA is when you have bases on top of each other, which is what happens in double-stranded DNA. If you remember from organic chemistry, when you have rings of carbon stacked on each other, that's very favorable. Pi stacking, more or less. So you have base stacking that's helping to put the DNA together. Let me go back to my PowerPoint here. Oh no, all my writing went away. So repelling, negative charge, attraction, hydrogen bonds, base stacking. And temperature is a measure of energy. So the higher the temperature, the more energy you have. So what salt does is that the salt, draw my DNA again, the salt is going to interact with all the negative charge. So when you add salt, and actually the DNA inside your body is just covered with ions, it's covered with magnesium. And what the salt does is interacts with the negative charge. And what that is called is screening or shielding. The strands of DNA are interacting with the positive charges. So they are no longer like repelling each other because they're kind of hidden from each other. That positive ion kind of neutralizes the negative charge. It doesn't actually neutralize it. It more or less hides it, is a better way. It shields it from other negative charges. So that's why if I add a lot of salt, I'm basically, so salt will reduce the repelling effect of the negative charge. Therefore, if I want to separate it, I have to put in more energy or increase the temperature more. So this question really gets at, do you understand the forces of DNA? Do you understand what's holding DNA together? And do you also understand what's trying to rip DNA apart? Because DNA is negatively charged. So the DNA inside your cell, it's just not always like super happy because of that negative charge. But you have a lot of magnesium inside your cells. So that magnesium is screening that negative charge. So yeah, any questions about that idea of the biomolecular forces in DNA? All right. No, we can move on. Again, if you're typing something out, feel free to continue to type it and I can always go back. And that brings us to, I thought we just did that. One sec, sorry. Yeah, I have the same slide repeated here. So this is not 3.2. This should be 3.3. And that brings us to DNA being genetic information. And this was not always known. There's my pen. This person is McCarthy. What's being written here is mouse lives, mouse dies, mouse lives, mouse dies. All right, so in the early days of biochemistry / molecular cell biology, back in the earliest 20th century, people thought that proteins were the genetic information of the cell. And that actually makes a lot of sense if you do not understand what DNA looks like, which they didn't at the time. They thought proteins, which was genetic information, because proteins are incredibly varied. So to mimic, or the reason why life is so varied on Earth is because, oh, everybody has different proteins. That protein is dictating how organisms grow. However, a series of experiments in the 1930s and 1940s, and even earlier than that, started showing that actually this substance called DNA was the genetic information. So the experiment I'm showing here is by Avery, McLeod, and McCarthy. One of the seminal key experiments, not the only experiment, but one of the key experiments that showed DNA was the carrier of genetic information. And they used two strands of bacteria, rough strand and smooth strand, right? And for this experiment, if you give a mouse a shot of rough strand virus, the mouse is fine, the mouse lives. If you give that mouse smooth strand, it dies. All right. And what they saw was if you take your smooth strand bacteria, they die as well. Or sorry, they live. So you take your smooth strand and you add heat to it, you kill all the bacteria. That's what I had in my head. You kill the bacteria and the mouse lives. However, if you take your smooth strand bacteria, you kill it. Then you add your rough strand bacteria and give that to a mouse, it dies. And what they were able to find out is that it was the DNA that was leaving the smooth strand bacteria, and the rough strand was taking up that DNA and transforming themselves into smooth strand. So basically the non-virulent bacteria would take up DNA and become virulent. So this was one key experiment that showed that it was DNA. And the reason they found out it was DNA is because they were able to label DNA in proteins. And they saw, oh, the bacteria are taking up DNA, but they're not taking up protein. So the reason why they're transforming is because of nucleotides. Therefore, DNA is the carrier of genetic information. So that was the key experiment by Avery, McLeod, and McCarthy in 1944. And since this is just a overview, I am skipping over a lot of other experiments that showed this as well. But this is just one of the big seminal ones. So now that we know the structure of DNA, now that we know that it is the genetic information, we understand how it replicates because of Chargaff's rule. Remember, A equals T, G equals C. This means that when we're replicating DNA, each strand is a template. And so DNA is said to replicate semi-conservatively. This means that each new DNA an organism makes is made from one old strand, also called a parent, and one new strand, called a daughter. So when we do replication, it's always semi-conservative. This allows to create two new strands of DNA that are more or less exactly the same as the original DNA. This DNA allows us to encode for proteins. This is called transcription. So DNA gets transcribed to mRNAs. These mRNAs are read by tRNAs, and we translate that into proteins. And this is called the central dogma of biology. DNA replicates, it gets transcribed into RNA and translates into proteins. Will there be essay questions on our exams? There will be short answer questions, yes. Essay, probably not. A lot of short answers, though. Right, so that is the central dogma: replication, transcription, translation. And let's get into sequencing. How do we read DNA sequences in organisms? And before we do that, let's start by talking about endonucleases, or restriction endonucleases. And what a restriction endonuclease is that it is a enzyme that cuts DNA. And the types we use inside the lab are cut at very specific sequences. And there's two types, endo and exo. Endo cuts within a nucleotide strand. Exo cuts like the last residues of a nucleotide strand. So usually we use endonucleases inside the lab to cut DNA. There's two types of, or two classes, type 1 and type 2. Type 1 cuts at some random site that's not the recognition site, which is not good for us. Type 2 cuts at the recognition site, which is useful. So inside the lab, what you will use the majority of the time are type 2 endonucleases. Here on the table, we have just examples of endonucleases. You don't need to know this, or you don't need to memorize it. Just know how to like read this table. So let's start with the enzyme name. These enzymes, they're named systematically, where the first letters tell you what organism it comes from, and the Roman numeral tells you the order it was discovered. So ECO-R means E. coli R strand. So ECO-R1 is the first one developed or discovered in E. coli R strand, ECO-R2, ECO-R5, so on and so forth. The recognition sequence tells you where it's going to cut, and the arrow tells you where in that sequence it will cut. And so we have two different types of cuts we can make, called a sticky or blunt. So when you make a cut, you either make a sticky end or a blunt end. And I have examples here of a sticky end and a blunt end for ECO-R1, ECO-R5. What a sticky end means is that when you make a cut, you have an overhang. So here we are making a cut. And so when we cut this, our resulting strand will be G, and then this will be C, T, T, A, A. This TTAA has nothing to hydrogen bond to. So that's why it's called sticky. It's looking to hydrogen bond to something. And the other strand would be A, T, T, C, G. These A, T, T, they have nothing to hydrogen bond to either. So they're looking to hydrogen bond. So this, you have created two sticky ends. For ECO-RV or ECO-R5, when we make a cut here, one strand is going to be GAT, oops, sorry about that, CTA. Everything is hydrogen bonded to each other. So there's no overhang. So that's why it's called blunt. And your other strand is going to be ATC, TAG. Again, no overhang. When looking at this table, blunt cuts happen when you cut and your nucleotides are even on each side of the cut. So for ALU1, 2 nucleotides to the left of the arrow, 2 to the right. equal amounts on each side, that's blunt. While BAM H1, that's going to be sticky. 1 on the left, 5 on the right. If you also make a cut at either end, that's also going to be blunt. So that's how you can tell blunt from a sticky, just based on the cut. Is it at the termini or is it directly in the center? You're going to make a blunt. If it's not in the center of the termini, it's going to be a sticky end. Usually you want to use sticky ends in the lab because the reason why you make these cuts a lot of the times is because of like PCR or you're doing some kind of manipulation of DNA. And so it's a lot easier to work with. But blunt does have its own uses as well. So you're not always going to use sticky end. Sometimes you will use a blunt endonuclease. But any questions about that terminology? Okay. So here I have a sequence for you. And I want you to figure out with this sequence, if I mix it with TAC1, what would be my new products? And then once you have that, just go through this table and see if you understand which enzymes would make a sticky end and which one will make a blunt end. So I'll give people a few minutes to work through this. As always, let me know if you have questions. All right, so let's go through this. So 4A, we're cutting this with TAC1. We go to our table, TAC1, is looking for the sequence TCGA. And this arrow says we're going to cut between the T and the C. One thing to note about this is that it always cuts 5' to 3'. So we're looking for 5' to 3' TCGA. There it is. So we're going to make a cut right there. And 5' to 3' going the other way. Remember, always read 5' to 3'. So here is TCGA. We're going to make a cut between the T and the C. So we're going to make cuts like that between this T and the C and this T and the C. So our resulting strands are going to be ACGT, then TGCA, GC. So that is sequence 1. And the other sequence is going to be CGAATC, then TTAG. So that's going to be strand 2. So that's how we're going to make our cut with tack 1. And then B, deciding if something's a sticky or a blunt end. Remember, if it cuts and it's on equal proportions when it's cutting, so 2 on each side, so that's blunt. If it's not, it's sticky, so sticky, sticky, sticky. If it cuts at the termini, that's blunt. Here, 3 on each side, blunt, sticky, blunt, sticky, 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 sticky. All right. So any questions about our restriction endonucleases at this point? Would PVUT bleed? Oh yeah, I think I was just getting a little sticky happy there. Yes, PVU2, very good. That is blunt because it's 3 on each side. All right. Now, when you cut DNA, one of the things that's often done is you run that DNA on a gel. And when you run DNA on the gel, here on the right, on the left rather, is our gel apparatus. And gels are always going to be like this, where you have a, you have a positive termini on the bottom and a negative termini at the top. And you're going to run it, and the DNA is going to go towards the positive termini. And the smaller the DNA is, the further it will travel down the gel. And so this is a way you can do after cutting your DNA. You can see what sizes you have. So usually you run a ladder that will be a known molecular weight in 1 well. And then when you do cuts, you can see what size fragments you have. So that's a very common biochemistry technique where you make cuts like this. And then you run it on a gel. Now, what I'm going to get into for the rest of the time today, and then probably continue on Wednesday, is how do you sequence DNA, right? And we're going to start with a historical method that's still used today. And then we're going to get into the high throughput techniques, most likely on Wednesday. So we start with the Sanger technique. Sanger is a very famous name in biochemistry. He's one of the few people to win 2 Nobel Prizes. So pretty smart. And his technique for sequencing DNA is shown here. This is called chain terminator sequence. And so what you do is that, let's say you have DNA that you want to sequence. The first step is to cut it with restriction endonucleases to make it into small fragments. Then you make it single-stranded, right? So you make your single-stranded DNA. Then you use his technique. So this template, what's showing here is, this is what we want to sequence. And so we start with a primer, and then we add a bunch of nucleotides. We have our regular nucleotides, ATP, CTP, GTP, TTP. This D stands for deoxy. So we have DNA, ATP, DNA, CTP, so on and so forth. We also have these DD dinucleotides, which are being shown here. DD dinucleotides are missing the OH at the 2' position like normal, but it's also missing OH at the 3' position. Now remember, when we're adding DNA, we always add them 5' to 3'. If we want to add new DNA, we need an OH at this 3' position. So if you take the OH away from the 3' position, you cannot add any more DNA to it. And also to these DD nucleotides, we have fluorophores. We have them labeled by dyes so we can see what color they are. And the idea here is that you make, let's say you make 1,000 copies of your template. And then you add all these nucleotides to all these different templates. And every time a DD DNA is added, you can no longer add any more nucleotides to it. And so if you have enough templates, what will happen by chance is that you'll stop at different points in different reactions. So if I have like 1,000 templates, let's say like run number 50 stopped here, run number 20 stopped here, run number 112 stopped here, so on and so forth. You can then take all these sequences and run them through gel electrophoresis to separate these DNAs by size. And they're always going to be separated by one nucleotide. You can then shoot the DNA with a laser and see what color it was. So here, what this image is showing, this is electrophoresis in a gel. So we're separating DNA by size here. And so you can see that our smallest DNA is blue, which means that, okay, right after our primer, it's blue. This means that there's a C there. Then we go to the next smallest nucleotide, that's also blue, okay? So that's C there as well. So on and so forth. Next smallest nucleotides, green, red, blue, yellow, red, yellow, green. So with this method, you can sequence small DNA segments, like 300, 500 nucleotides long. This sequence is, this method is great at doing that. And it's still used in labs today. Like I used this when I was doing lab work, less than a decade ago now. But any questions about the logic I just went over in this chain terminator sequence method. Yeah, so I'll do one more slide here because it relates to the chain terminator sequence. And so what happens when you want to sequence small amounts of DNA? You send your DNA to a lab that specializes in doing this method. And they can do this really quick, like in less than a day. And then you'll get an email back that looks exactly like this image. And this image is your DNA sequence. What each peak means is basically like how confident we are that your sequence is this nucleotide. So you can see when looking through this image, like you only see one color a peak each time, which means that like at every position, we're confident that the nucleotide we're seeing is there. Mainly at this position, there's you can see other nucleotides kind of pop up, but it's getting dwarfed by your A here. As you move further down here, if I would show you like 600 or 700 nucleotides down this line, you'll start to see all four colors being the same because due to the chain terminator sequence, it's only good for a short amount of nucleotides. These numbers are telling you the sequence number. So it goes from 1 to whatever DNA you give. And this is really a way to not only sequence DNA, but in working in the lab to make sure that the DNA you're working with is the sequence you thought. So a common thing to do is once you get this back is you read it by hand to see if it matches up. So for example, if in the lab I was interested in positions 130 to 150 and the sequence was here, I would go by hand and just say, okay, is that the sequence I expect? So the sequence I expect should go A, G, C, T, C, C, A, G, C, T, 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 T. Remember, all I'm doing is reading this and seeing if it matches up. Then it should go C, and I found an error in my actual DNA sequence. So in my DNA sequence that I expected, I thought there would be a C at this position. When I got my results back from the lab, there was a G at that position, which means a mutation in my experiment has happened that I didn't want. So it's very important when you're working with DNA, before you do any real experiments, you should really determine what the sequence is. Otherwise, you're going to spend months of your time and then you might remember, oh, I should probably sequence that. And then whoops, all the experiments I did were meaningless because I had an error in my DNA. And you can do this easily enough through the Sanger sequencing method. All right. Any questions about the chain terminator sequence method here? All right, if not, that's the end of class for today. Thank you all for coming, participating, listening. I appreciate it. I will grade your homework for that was due this morning. I will put up a new one. Remember, you have a week to do it. If you have any questions, let me know. Otherwise, hope you have a good rest of your Monday, and I will see everybody on Wednesday. Take care, everybody.